Hello dear students. In this lecture, we will discuss about the different techniques for microbial analysis of air. So here there are several methods which require a special devices specially designed for the enumeration of microorganisms which are present in the air. So the most important one are nothing but the solid impingement method and liquid impingement uh, devices, or we can say a method. Uh, Solid and liquid impingement devices, filtration, sedimentation, centrifugation, electrostatic precipitation. These are nothing but the different methods which are used for the enumeration of microbes which are present in the air. However, none of these devices collect and count all microorganisms in the air sample which are being tested by using these methods. Some microbial cells are destroyed and some entirely pass through in all these processes. Some solid and liquid impingement methods or devices we will discuss in this according to your syllabus. So here we have to discuss about the solid impingement devices and liquid impingement devices. So here firstly we'll discuss about the what is meant by the solid impingement devices and what are the different types. In case of solid impingement method or devices, in this method, the microorganisms are impeached, or we can say, in other words, collected or get trapped in the devices, get directly trapped on the solid surface which is used in these devices on the agar medium. The agar medium is incubated until visible colonies appear on that surface of that agar medium. So here in these solid impingement devices, special devices is designed so that the in that devices a solid already sterile media we have to place and air has to be passed so that the suspended dust particle or microorganisms get trapped or collected, and so that as the availability of nutrition they start grow on the surface of that solid media. And when we incubate that particular plate, we have to incubate until the visible growth of the organisms are found or appear on the surface of this solid media. And we have to count that particular colony so that we can get a total approximate number of different numbers of organisms which are present in the air. It is assumed that only one colony is derived from a single microorganisms entrapped on the surface of the agar medium. Thus, number of microorganisms can easily counted by counting the number of colonies in given volume of air sample. So, there are various types of solid impingement devices uh, or methods. We can say devices are discussed in your syllabus. These are nothing but the settling plate method. It is the most important method of solid impingement devices. Another is funnel device, sieve type of sampler. This is another third category of solid impingement device. Slit type of sampler, Anderson air sampler, and membrane filter technique. So another membrane filter technique we have already discussed in water bacteriological analysis of water. So here to also discuss about the organisms or enumerate the number of microorganisms present in the air, we will use different solid impingement devices. So firstly, we'll discuss about the what is meant by the settling plate method or settling plate device. This is the simplest method of enumeration of microbes in the air. In this method, the cover of the petri dish containing an agar medium is removed and the agar surface is exposed to the air for several minutes. So this is nothing but the we have to prepare a simple nutrient agar plate, and after sterilization uh, where we have to enumerate the number of microorganisms, whether it is indoor or outdoor environment. We have to simply remove the upper lid of your petri plate, and the media which is present in that plate we have to expose to the air so that the organisms which are present in the air get trapped on the plate, and after incubation they forms their visible colonies on that medium. And the agar surface is exposed to the air for several minutes. After exposure, the petri dish is closed and incubated for 37 degrees Celsius for 24 hours, and it is the optimum condition for the growth of organisms. After that, certain number of colonies which are of representative growth of particular organism which is present in the air start developing on the surface of the agar medium in that petri plate or petri dish. Each colony represent the single type of microorganism in that air. So on the basis of morphological characteristics or cultural characteristics of the particular colony, the differentiating colonies we have to count, and on the basis of that, those different colonies we can observe, we have to count only once. This is not a correct technique of sampling of air because all microorganisms do not settle on the plate, and also the amount or volume of the air sample is not known, as we are directly exposing that particular nutrient agar plate into the environment. So volume of the air sample we didn't know, so that we could not count the total number of count with respect to the volume of air. It gives only a rough estimate. This type of technique is used in industries to know the microbial content in the aseptic room. So while discussing about the aseptic condition, we can also study aseptic condition is nothing but the that particular environment which can maintain in the particular industrial area or aseptic industrial aseptic room, or we can other terms we can also call it nothing but the operation theater which can be used in the different hospitals. So this particular environment is aseptic. That means there is no forms of different microorganisms they are removed from that particular environment. So that particular direct settling plate technique can be useful to determine the or maintaining the particular condition of that aseptic condition of that room. So whether that air is contaminated or not. So we can, for that particular environment, we can use that settling plate technique only. Otherwise, in the outdoor environment, we could not justify that settling plate technique with respect to, we could not determine the volume of air or the number of microorganisms which are present in the air. So these are nothing but the limitation of settling plate technique. Next technique we'll discuss in next lecture.